The project by Christian Althaus and Emma Hodcroft, from the University Bern, and Richard Neyer, from the University Basel, has combined two different approaches, namely phylogenetics and epidemiological modeling, to study the virus spread.
The large number of viral sequences that were collected around the world during the SARS-CoV-2 pandemic provide, for the first time, unique insights into how different variants of the virus spread between people and across countries. This inspired us to integrate this knowledge into mathematical models that can be used to study the impact of control interventions, such as surveillance of travelers, and to inform policymaking during the pandemic response in Switzerland.
To do this, the researchers used NextStrain, an open-source platform that provides tools and visualizations for the analysis and interpretation of genetic data.
NextStrain uses the viral genetic sequence to uncover how the virus spreads and changes through time. This technique, known as phylogenetics, helps us to show how SARS-CoV-2 variants evolve and move around the world. So here, this map shows the spread of alpha at the end of 2020, beginning of 2021. It started in the UK, so you can see how originally it starts to move to other countries, but as it spreads, it then starts to move from countries within Europe to other countries within Europe. Figuring out how many times alpha or delta might have come into Switzerland is difficult. No one can count this directly. But using sequencing and phylogenetics, we can estimate how many times variants like alpha or delta have been introduced to Switzerland, and we can use this information to start our simulations. This helps us better understand what happened in real life, but also we can alter the simulations to see what might have been different if we'd had different measures in place.
These analyses were not only important for pure research purposes, but also for deriving measures to stop the pandemic.
Our project provided critical insights into the importation and early spread of SARS-CoV-2 variants in Switzerland. For example, we showed that border closures would have had a limited impact on the spread of alpha and delta in Switzerland, while increased surveillance of travelers can potentially delay the spread of new variants in Switzerland.
As viral sequences for other pathogens become increasingly available, Althaus and Hodcroft hope that the integration of phylogenetic analyses into epidemiological models will also be possible for other infectious diseases. This will allow informed decisions to be made about intervention strategies and shape future pandemic preparedness.